Thank you everybody for tuning into this special presentation about COVID-19 and how Addgene is responding to this pandemic. First of all, I'd like to echo what Joanne said and really just thank everyone, all of the Addgenies, for your hard work during this difficult time. And I'm delighted to be able to tell you a little bit more about what some of that work has come to so far. I'm just so proud that Addgene is stepping up and being part of the solution here, and it's something that you all should be very proud of as well. So this talk is divided into 3 parts. I'm going to start by giving you some of the background, and then I'm going to hand it over to Marcy, who will tell you more about Addgene's plasmid resource and some of our response and how we've been handling a lot of the deposits and requests during this time. And then Caroline will wrap up by talking about how Addgene is engaging the community and how the scientific community at large has really stepped up to this challenge as well. All right, so first of all, I'm going to take a page from Eric and talk about some definitions first. And so some people have probably heard COVID-19 or SARS-CoV-2, and what's the difference between these two things? So COVID-19 actually stands for coronavirus disease, and it was discovered in 2019, late 2019. And so that's the name of the disease, and that's actually caused by the virus SARS-CoV-2. And so if you go into the NIH GenBank, what you'll actually see is SARS-CoV-2 as the name of the virus. And one way you can think about this is like AIDS and HIV, where AIDS is the disease and HIV is the virus that actually causes that disease. A lot of you have probably also heard of SARS as well. So this was a disease that hit in the early 2000s, and it stands for severe acute respiratory syndrome. And the reason this is important is because SARS is caused by the virus SARS-CoV-1, which is 96% identical at the nucleotide level at the sequence level to SARS-CoV-2. And it's so similar that actually the WHO considered calling this disease SARS-2. And actually in a kind of an interesting branding story, they thought it would be too scary to call it SARS-2. So that's why they went with the COVID-19 name. Probably in retrospect, maybe it would have been better to have it a little bit more alarmist in the beginning. But I did think that was kind of an interesting story, how they came across these different names. And it is important that these are very similar because a lot of what we learned from SARS-CoV-1, people are using to build on that for the SARS-CoV-2 research. So they're all part of a class of viruses called the coronaviruses. And probably most of us have gotten a coronavirus in the past because they're responsible for about 10 to 20% of the common colds that we get. And there are 4 different types. So the alpha, beta, gamma, and delta. And the alpha and beta coronaviruses are caused through a zoonotic transmission, which is basically that a virus starts in one species and jumps to another one, and these start in bats. And the gamma delta start in birds. And so I'm not going to talk too much about those because those are not known to come to humans. But the ones from the bats is where the common cold viruses come from, and those are highlighted in this red box here. But more recently, we've seen more serious beta coronaviruses come through bats, and that's SARS, MERS, and now SARS-CoV-2. And the way that this works is it starts in bats, but it usually goes through an intermediate host. And so in the case of MERS, it went through camels, and in the case of SARS and now SARS-CoV-2, it's thought to have gone through civets or other small carnivores. And what happens is the virus undergoes small mutations that allow it to jump from one species to the next. And one really important thing about the coronavirus is it's thought that bats have maybe 5,000 different coronaviruses. So any of the research we're doing now will probably be applied later because scientists think it's quite likely that there will be other zoonotic transmissions of other coronaviruses from bats eventually to humans as well. So it's definitely something that's worth studying into the future. And coronaviruses get their name from their shape, their crown-like shape. And so this is an electron micrograph of what a coronavirus would look like. And you can see these characteristic spikes here. And these are caused by the spike protein. And a huge shout out, by the way, to our own Maya Kosman for making this image, which is now on the IGI website. Really beautiful work. And so what you can see here is the blue spike proteins, as well as a number of other proteins, which I'll talk a little bit about later. And this is what the coronavirus looks like. The spike proteins are very important because that's what gives the coronavirus entry into our cells. So this is what the genome looks like, the SARS-CoV-2 genome. And so one thing to note is it's an RNA virus, meaning that the RNA is what's packaged inside. And so we talked a little bit about HIV earlier. HIV is another great example of an RNA virus, whereas something like AAV or adeno-associated virus is a DNA virus where the DNA is packaged inside. And so coronavirus is RNA, and it's about 30,000 bases, 30,000 RNA letters. And this is what the genome looks like. It encodes a number of different proteins. For those who are interested, Marcy will talk a little bit about the non-structural proteins, but there's a little bit of in the appendix of more detail for some of these proteins as well. I'm just going to give you a really high level for now. And so the non-structural proteins and the accessory proteins, they're involved in viral replication, viral assembly, and decreasing the host defenses. So they're proteins that are involved in helping the coronavirus make more copies of itself. Whereas the structural proteins are what you actually would find in the virus itself. Those proteins are there. And so we talked a little bit about the spike protein, and there's also the envelope protein, the E, and the membrane protein, what you'll see is M there. And those make up the envelope of the virus. And then there's also the nucleocapsid protein, which is the N, and that's inside encapsulating the RNA as well. And so there's a lot of interest around these proteins because these are things that scientists could, for instance, use for antibody tests. They're actually what you would find in the virus. But these are also equally important for the virus to replicate. And so I'll tell you a little bit about the life cycle here. And so what we saw in the last slide was the viral genome and the viral proteins, but equally important are the host proteins. So viruses cannot replicate on their own. They need a host, whether that's a bat, a camel, a civet, a human, in order to make more copies of themselves. They don't have all the machinery that they need on their own. So it's very important for them to actually gain entry into a host. And in the case of humans, what we know is that they are binding to this ACE2 receptor, which probably many of you have heard about. And so the spike protein will interact with the ACE2 receptor and allow the virus to get into the human cells. And once inside, the RNA will become unpackaged and be in the cell. So this is that RNA genome, kind of a smaller version of what I showed you in the previous slide. And this is what scientists are actually looking for when they're doing, say, people talk about testing, right? The PCR test. What they're actually doing is they know the sequence of this, and they are looking inside people to see whether or not these sequences are present. And so when you get infected with the virus and the virus is replicating, there's going to be copies of this. And then as you're getting better, that load is going to go down. And so once you're clear of the virus, then you can come out of quarantine when you no longer have copies of this circulating. Okay, so that's the genome. Now we're using the host machinery to start some translation, so meaning that it's being turned into protein. And these are those non-structural proteins I talked about earlier. They're going to help the virus make more copies. So in this case, they're calling it replicase. What this is it's an RNA-dependent RNA polymerase, where it's going to be able to take the RNA and make more copies of the virus. From here, it's going to make the structural proteins, which you'll see here, and then that RNA is going to get packaged, and basically more viral particles are going to be made through this cycle. And so this is a combination of the proteins from the virus and proteins from the host. And it's really important that we understand this biology, because this is how scientists can start thinking about therapies. And so one example of this is remdesivir. And so remdesivir is thought to act by inhibiting this replicase, or this RNA-dependent RNA polymerase. And you can see the logic behind this. If we could prevent the virus from making more copies of its RNA genome, this would be one way you could stop the virus from replicating and making more copies of itself and spreading. And scientists are looking at drugs all along this pathway, both at viral proteins and at host proteins, in order to see how we could stop the virus from spreading and replicating. So just kind of to bring it all together, scientists have, they really need plasmids both for the viral genome, the SARS-CoV-2, and for the relevant host genes. This will allow us to understand the basic biology for SARS-CoV-2, which is really important for this pandemic, but also really looking forward to how we can prevent future outbreaks or get them under control much more quickly by really understanding that biology of how coronaviruses work. Using plasmids, you can also start to think about diagnostics. We talked a little bit about qPCR and the sequences there. I didn't talk too much about antibodies, but again, qPCR detects active infections. And then once you've been infected with something, your body's going to make antibodies against it. And what it does is it makes antibodies against those structural proteins that we talked about, right? So for labs to have access to plasmids that express structural proteins will allow them to start thinking about how to make antibody tests. Also for developing vaccines, scientists are using RNA and protein sequences from SARS-CoV-2. And also we talked a little bit about therapies and how, having these proteins available to scientists will allow them to see which drugs can interact with them and which proteins could, for instance, like their activity could be inhibited by these drugs. It's just so important to have this toolkit available to scientists. And so Marcy is going to tell you a little bit more about Addgene's specific collection of plasmids, how we're handling those, and give you some examples of how scientists are able to use those plasmids.
Great, thanks, Melina. So as Melina mentioned, scientists can use plasmids for understanding viral biology, detection, and development of therapeutics, which leads to the obvious question, what exactly does Addgene have available to support these efforts? I'm happy to report we have a lot of things available. We have over 100 plasmids that are online that are not related to the Ginkgo collection, and as of today, we've managed to put 383 Ginkgo collection plasmids online. Which is a really great resource for scientists studying the SARS-CoV-2 virus. The plasmids we have really run the gamut across all 29 viral components that Melina described, as well as a variety of already known human targets. And the reason these human targets are already known is because of the previous research into the SARS-CoV-1 virus. Thinking back to Melina's introduction, you know that the spike protein as well as the ACE2 receptor were two really important components of the virus host cell interaction. So it's no surprise that we have a large abundance of both of these types of plasmids represented in our collection. We also have a set of plasmid tools that can be used in some of those detection experiments that Melina described. So some of these tools are brand new. Scientists really were able to ramp up their production of specific enzymes used in some of these detection assays, as well as think about older tools that were previously created for something else, but could be utilized for detection of the SARS-CoV-2 virus. So two great examples of CRISPR-based tools that Addgene already had in our collection were the SHERLOCK and DETECTR systems. And both of these now have SARS-CoV-2 specific protocols created so scientists can use these tools as they're trying to detect when people have gotten the viral infection. So that's a really general overview of what we have. The next thing I want to do is describe Addgenie's efforts in getting these plasmids into our repository and then quickly making them available to scientists doing this frontline research. So as everybody knows, Addgene shut down along with most rest of the world, including many labs doing research. And then over time, as we identified essential groups trying to move research forward in order to combat COVID-19, Addgene knew that we were in a great position to help these scientists by making research reagents available to them quickly. So our goal with the first sets of deposits was to really maximize our turnaround time, even if it was slightly more resource intensive. This allowed us to seed our collection with a variety of useful plasmids that allowed frontline researchers to really jumpstart their research and make sure things were moving forward really quickly. Because we weren't doing any other processing of deposits in our lab, we could give these deposits real white glove treatment. We did a lot of manual processing. We held their hand all the way through from start to finish. And we also kind of double dipped in our QC. So for people who watched the NGS talk last week, you know that Sanger sequencing is a very fast way to turn around QC, but it does give us very limited amount of data related to the plasmid sequence. Whereas next generation sequencing, or NGS, is a slower process, but it allows us to verify the entire plasmid. So for these first early deposits, we made the decision to perform Sanger QC, which allowed us to release these plasmids very quickly, and then followed up with a second round of QC at a slower pace that allowed us to verify the entire plasmid. And I do want to take this moment to shout out the lab and QC teams for this quick turnaround here. From receipt to the plasmids becoming available for request was about 1 week. And to put that into context, it usually takes us closer to 5 weeks to turn deposits around. So that was a real heroic effort on their part. Of course, white glove treatment is not a sustainable process. So after we got a good solid base of plasmids, we wanted to switch over to a more tenable way of processing the incoming deposits. So where we're at right now is really looking to expand and diversify our selection of research tools. And doing so, we want to be able to utilize more of our standard high throughput lab processes. So right now our processes look much closer to our normal intake process, but because we still want to make sure we prioritize the COVID-19 plasmids, there is a lot of additional energy and oversight that the lab is doing to ensure a rapid turnaround time for those plasmids. And within the last few weeks, we have started processing non-COVID-19 plasmids as well. So we're making sure that non-COVID, non-SARS-CoV-2 research materials are also getting processed, but at a slightly lower priority. As many of you may have heard, we are really excited about this Ginkgo collection. Ginkgo is synthesizing a ton of SARS-CoV-2 constructs to really help out the scientific community. This collection is huge, and when I say huge, I mean on the order of 3,200 plus plasmids. And to put that in context, most deposits are anywhere from a few, like 2 or 3 plasmids, to maybe a couple dozen. So this is 100 to 1,000 times bigger than some of the plasmids that we've been receiving. So therefore, we need to treat it differently than our standard deposits or even our standard COVID-19 related deposits, just due to the fact that it's so huge. That being said, we're really excited to be working with Ginkgo on this effort. This is a really important and versatile collection. They've done a great job in identifying the types of plasmids that scientists will use and created it in such a way that there's really something for everybody. There are viral parts with a variety of tags, so they can be applied slightly differently in different experimental settings, and in a variety of backbones that allow scientists to work in yeast or E. coli or various mammalian cells, whatever type of system they have up and running in their lab. Of course, we still want to make sure we're balancing our quick turnaround time with efficient resource use, and now doing this for a very large collection. So to that end, Ginkgo has very generously offered to do some of the next generation sequencing for us. And this is incredibly generous, but it's also completely unprecedented from Addgene's perspective. We've always done our own QC and applied our own results. So it's taken a lot of logistical maneuvering to make sure that we were able to take Ginkgo up on this very generous offer. And thanks to the Curation Lab, QC, and data teams for their flexibility and willingness to try something new. This is something that we're moving forward with in the next few weeks. It's still a little bit of a work in progress, but things are moving forward really well, and I'm really excited that we're able to do this. So we have plasmids. How do we get them out to scientists? What many people may not quite appreciate is there are a lot of scientists who are not able to go to their labs and do research, but they're still placing orders so that they'll be ready to go when their labs reopen. So the big question is, how can we ensure that we are only shipping plasmids out to customers who are ready for us to do so? And additionally, how can we make sure the COVID research are getting their stuff first? And the answer to that question is really a daily logistical ballet performed by CSI support, lab, and shipping. So outlined here is the process these teams go through every day to make sure the right things are going out to the right people at the right time. So we're emailing every customer. As of yesterday, over 3,500 emails have been sent out. Once a customer confirms that they can receive their order, CS or shipping ops marks these orders as high priority, and if it's for COVID-related research, they'll add an additional note. And the high priority label plus that additional note allows the lab to really prioritize their picking batch and gives the shipping team information as to what shipments are highest priority to make sure that they go out. And in addition to the teams who are intimately involved in this process every day, I do want to shout out the product and dev teams for creating this high priority tagging system and for their ongoing efforts to really make this process more automated for our staff. So Addgene has done our part. We've taken plasmids in. We're doing a great job getting these materials out to scientists. So the next question is, what is it that researchers can do with these plasmids? So as Melina mentioned, there are a lot of things that people don't know about the basic biology of this virus. It's novel. This is the first time we've seen it. So there are some basic questions as to what the different components do. And you don't need to worry too much about this list. It's quite technical, but what I do want to highlight is even within the known viral components, there are a handful that we have no idea what their function is, and therefore we don't know how we could potentially target that protein or what effect it might have. So in addition, there are some things we already know. For example, we know the spike protein interacts with the human ACE2 receptor. So using some of the plasmids, we can take a much closer look at this interaction and potentially identify very specific places that we can target and disrupt this interaction and potentially keep the virus from infecting our cells. One thing that we can also do is use our knowledge of the viral life cycle to see if we can stop it in its tracks. So remembering back to Melina's slide on the viral life cycle, we know that the virus needs to bind the ACE2 receptor in order to get into a cell. So one of the questions scientists are asking is, can we find a version of ACE2 that binds the virus better than our normal receptor does? And the answer to that is, I'm happy to say yes. If you look at the dots in this green box, those dots represent different versions of ACE2 that bind the virus better than the normal receptor. So what can we do with this? Well, it's possible we can create a therapeutic decoy with one of these high binding versions of ACE2 that, if administered correctly, could soak up the virus and keep it from binding into a human cell, and therefore it's a dead end. The virus can't replicate. We can also look more globally at what the virus is doing with host proteins. So we know that a lot of host proteins are involved with the viral life cycle. The virus basically hijacks a host cell in order to replicate itself and create more disease. So one thing that we can do is do a global study and see what host proteins are involved with the life cycle. And then we can further look and see if there are any host proteins that already have an FDA-approved drug that acts on them, with the idea that we can really jumpstart some therapeutic efforts by making something that's already on the market and proven safe and use it in a new context to really help people suffering from COVID-19. And I'm happy to report that the viral vectors team is making some lentiviral preps of some of these types of plasmids that will really help scientists jumpstart these studies. So doing these types of studies is really labor intensive, as creating virus takes days to weeks of work in the lab. So the fact that we'll have some ready-made preps available will save scientists tons of time and money. And I just really want to end here noting how much effort our efforts are making a difference. So as Joanne mentioned, we've had a lot of requests for these plasmids, and they're flying out the door. In the few weeks we've had these plasmids available, we have tons of requests. Most of them are flame, most of them are blue flame plasmids, which really means we're getting a lot of plasmids out into the community so scientists can further our efforts in fighting this, this virus and this disease. And with that, I'll turn it over to Caroline, who will talk a little bit more about the community engagement.
Thank you, Marcy and Melina, for that great info on the coronavirus and the plasmids in our collection and all the amazing distribution we're doing. So next, I'm going to talk a bit about what we see happening in the scientific community during this pandemic. So how are scientists and Addgenies responding, and the impact that all of us are having. Scientists in our community have really sprung into action in this crisis. They see problems and they apply their expertise to solve them. We can see this happening in real time in conversations taking place on Twitter and the emails that are coming into our inboxes. And here at Addgene, we're no different. Our Addgenies are finding ways to listen to what scientists are talking about, to understand their needs and problems, and to use our expertise to respond and offer solutions. Over the next 3 slides, I'll show you some examples of how scientists and Addgenies are responding during this crisis. This first example relates to how Addgenies communicate about our operations externally. As labs and businesses have begun shutting down during this pandemic, scientists who are continuing to work on pressing issues such as this coronavirus and the drug discovery need to know, where can we reliably get our reagents? So as you can see here, Addgene's Marcom, customer support, scientific support, and shipping teams are all working together diligently to make it clear on our website that we are open to let scientists know what we're shipping and to inform scientists about how they can speed up their orders and the process. So what you see here is the red bar called our site-wide banner, which We update with the most pertinent information about Addgene being open and our shipping logistics, and link to some more information for scientists. One of these links goes to our COVID-19 updates page. We keep this page updated with the most current information on our shipping and logistics. This has information and instructions for how to place shipments on hold, how to prioritize COVID-19 research-related orders, and more. What we then do is take all of this information and make sure that our support teams have it so that they can update their emails and their responses to customers, as well as our social media team having this information so that they can update our Twitter, LinkedIn, and other social profiles and postings so that wherever scientists are looking, they can know that we're open and we're here and ready to help. It's interesting to note that since we launched this COVID-19 updates page on March 12th, it's had over 5,000 views. So our community is finding it, and hopefully they're finding the plasmids they need and ordering. Then there's the new content that we're creating in response to the needs of our scientists. For example, in response to reports that the SARS-CoV-2 diagnostic testing was being slowed down due to a lack of RNA extraction kits, we started to see that scientists were talking on Twitter about the various things they could do to help. So they were crowdsourcing unused QIAGEN RNA extraction kits from closed down academic labs and getting those to public health testing facilities. Scientists were also talking about sharing protocols for nucleic acid extraction that would not rely on these kits. So what did Addgene do? We saw this and we said, hey, we have expertise here. We have expertise on DIY methods for DNA purification, which you see in the second tweet. Beth quickly published a blog post describing these methods. This information in this particular tweet was liked over 80 times, and scientists were glad we put it out there. But they did ask the next logical question, which was, you know, what we really need is an RNA extraction protocol, since this coronavirus is detected via RNA. So Leah quickly brushed up on her knowledge and published a kit-free RNA extraction protocol. It's shared openly on our website and as seen here on Protocols.io, which it's awesome to know that it's been viewed over 200 times in less than 3 weeks. My last example relates to our industry distribution. As Addgene has helped make more SARS-CoV-2 plasmids available, we quickly had lots of requests coming in, as well as, some complaints or concerns from scientists regarding the access restriction for industry and commercial scientists. So in this tweet, it shows scientists' discussion on Twitter regarding the SARS-CoV-2 gateway entry clones deposited by the Fritz Roth lab. So at Addgene, we saw these concerns, and we quickly prioritized them. So Matt Takborian quickly got in touch with the depositors and tech transfer offices to see if we could help them make these plasmids available to industry. This involved listening to both what the scientists needed from us, which was broader access, and listening to the concerns of the tech transfer offices. So Matt even worked with them to make customized solutions for the IMTA for COVID-19 research specifically, which is really awesome. We also quickly worked to make these plasmids visible on our website to industry scientists. So in this example table, you'll see we now add columns to our plasmid tables that indicate whether a plasmid is available to scientists in industry. Because during this pandemic, we know not just academic scientists are doing the important research to find tests and solutions. We know that pharmaceutical companies, biotechs, and other commercial labs are working on this problem. We've also at Addgene been working with the search teams to improve how plasmids available to industry appear in our search results, again, to make this more visible to those new potential Addgene requesters. To date, we've had 3 industry requests of COVID-19 related plasmids that have shipped out, and we look forward to more of those as we continue to promote that these are available. So as these examples really show, COVID-19 has had an impact on how Addgene does our external communications on our website. This crisis and this pandemic has had an impact on the content that Addgene is putting out in response to the needs of our scientists, as well as it has had an impact on the rapid expansion of our industry distribution and growth of that collection, of which I've talked about in the last few slides. Another thing we've seen during this pandemic is that COVID-19 and Addgene have had huge impacts on science. First, we've seen more broad sharing of reagents, data, and publications, including preprints, which, as a bit of a definition, a preprint is generally an article similar to what you would see in an academic or a research publication, but it generally hasn't gone through the peer review process yet, so it can come out a little quicker and will usually go on to peer review and full publication. So if you want to know more about how COVID-19 has impacted this type of open sharing in science, I recommend reading the Nature Biotechnology editorial that's linked in this slide deck. More organizations and companies we've seen in this COVID-19 response are quickly building new tools and opening them up for sharing. So the Ginkgo SARS-CoV-2 plasmid collection that Marcy talked about is a great example of this. There's going to be over 3,200 plasmids available. The first 384 just went online today through Addgene. And these are available to all scientists under the open MTA, which is really exciting and shows how companies, even such large companies as Ginkgo, are responding in this crisis. And then really, COVID-19 has had a huge impact on the new research data, publications, and findings that are coming out about this coronavirus, and really adding to the global knowledge of this virus and this pandemic, as well as future viruses. An example of the power of data sharing is shown here. And this phylogeny is based on full sequence analysis. It's listed on nextstrain.org. And it's showing the evolutionary relationship of SARS-CoV-2 viruses from the COVID-19 pandemic. What you see here is how scientists around the world have been able to sequence the virus from patients and then share this data with others to quickly understand the spread and transmission of the disease. And we've seen Addgene's impact during this pandemic in some of the newest research findings and articles that are coming out. I'll just describe 3 here, though I'm sure there are many more. The first article here came out in late March, and it characterizes that spike glycoprotein that Melina and Marcy talked about. The authors used lentiviral plasmids from Addgene in their experiments, and they deposited new plasmids for overexpression of SARS-CoV-2 spike protein in mammalian cells, which are currently in QC, and we hope to have available soon. The second article listed here is a preprint out of bioRxiv. So again, this is newer and hasn't gone through peer review yet, but it has some interesting information in it. So scientists describe in it using RT-qPCR to assay to detect SARS-CoV-2, and they describe procedures for reagent production. The authors have deposited the plasmids from this paper, and this work is really neat because it suggested to use this as a detection system for labs that may be in low resource settings and don't have same access to the testing kits that may be available here. So Addgene's helping make those plasmids available. And then the last example is a preprint, also in bioRxiv, that came out just last week. And this is in which the lab used deposited plasmids that they had made in a previous article and made available through Addgene. And they used these plasmids to develop a CRISPR-based tool to detect nucleic acids. So what's interesting here is this article started as the authors testing for influenza strains, so of the flu. But they actually expanded their test to test for SARS-CoV-2 once this pandemic started. So it's really, all of these are interesting examples of how the pandemic is having an impact on research in real time. And I really hope you can see through this whole talk the amazing impact that scientists and Addgene and all of you Addgenies are having during this. With that, we have to thank all of you. All of you have had a huge impact on the science being done now to understand coronaviruses and the COVID-19 disease. And really, you should all, find some way to take a break or give yourself a pat in the back. What you do really makes an impact in science. So thank you.